Okay, welcome students to the class of molecular cell biology. And today we will be talking about cell cycle and its molecular events. We have already talked about the fundamentals of cell cycle, what it is, what are the different events which go on during the entire cell cycle, and what is its significance and important to living cells, right? So today we'll carry on with the molecular aspects that regulate the cell cycle and how each and every event in the cell cycle is coordinated by the accumulation and deaccumulation of various regulators of cell cycle, which are different types of proteins and protein kinases. So let's begin with the lecture. So I'll just again, first of all, introduce you to the cell cycle in brief and what is the importance of all these molecular events to coordinate the fundamental role of a cell, that is to grow and divide, so that it can easily and successfully give rise to new daughter cells and carry on the process of life in the same way. So the timing and rates of cell division in different parts of an animal or plant are crucial for normal growth, development, and maintenance. This we have already discussed, that why this division and growth needs to be coordinated, correct, and why it is important for living systems. The frequency of cell division varies with cell types, right? It is different for each and every type of cell. There are some cells that divide very frequently, they divide very fast. For example, the cells of the immune system, as soon as a pathogen attacks, they divide very rapidly and the number is also very vast. But these cells, they divide only after they get a specific response or they encounter a pathogen. On the contrary, some human cells divide very frequently throughout life. That is the skin cells. You know, whenever there is a damage to your skin, or sometimes you can say for renewal also, you will see that these cells constantly divide. While others have the ability to divide, but they keep it reserved. For example, liver cells. You know, liver is one of your organs that it can regenerate on its own. This clearly indicates that these cells have the ability to divide again, but only when stimulated. So they keep it reserved unless and until some stimulus is there, some requirement is there, unless and until the liver cells are stimulated, right? Then only they divide. So they just keep it reserved. It is not like that randomly the liver decides that it is going to divide. Then there are, on the contrary, other cells like mature nerve and muscle cells that do not appear to divide at all after maturity. You know, if there is a damage to these tissues, it is considered to be a permanent damage. And these cells, they never, after maturity, they never regain the ability to divide, right? So the point is that in a single living system, there can be different type of cells with different types of division behaviors. Now, investigations of the molecular mechanisms regulating these differences provide important insights into the operation of normal cells and may also explain cancer cells' escape controls, right? If we learn about the molecular control of all these regulations that are being implemented for coordinated functioning of the cell cycle, we also get to know what are the points basically that are skipped by cancer cells that leads to uncontrolled growth and division of tissues. Now, I just mean to say various researchers, they have worked on the molecular control of cell cycle. And initially, all of them, what they could conclude was that there are cytoplasmic signals that actually drive the cycle. This means that they said there is something getting produced or accumulated in the cytoplasm of dividing cells that is missing when the cell is not dividing. right? So they termed commonly it to be the cytoplasmic signal. So it was clear with all the researchers that cytoplasmic signals actually drive the cell cycle. The cell cycle appears to be driven by specific chemical signals that are present in the cytoplasm. Some of the initial evidence for this hypothesis that these uh, drivers or regulators of cell cycle are present in the cytoplasm came from experiments which cultured mammalian cells at different phases of the cell cycle, and then these were fused to form a single cell with two nuclei. Further in my lecture, I'll also tell you about one of the very famous experiments, which is called the cell fusion experiment, where you will know how these experiments, they actually helped researchers to know that it is actually these cytoplasmic factors that drive the cell during their cell cycle from one phase to another. Fusion of an S phase cell and a G1 phase cell induces the G1 nucleus to start S phase. This suggests that chemicals present in the S phase nucleus stimulated the fused cell. Fusion of a cell in mitosis, that is the M phase, with one in interphase, even G1 phase induces the second cell to enter mitosis. So this is just an introduction to what you're going to know during this lecture about the molecular events of the cell cycle. We'll just see how these experiments were carried out. I'll tell you the exact mechanism of the molecular control, that is the maturation promoting factor control of the cell cycle. And in between, I'll also talk to you about the various important experiments that actually led to the discovery of this molecular factor. So any questions till now? Are you clear with what we are going to read in today's lecture or what we are going to learn in today's lecture? If you have a question, please type in chat. If you do not have a question, please type OK so that I can proceed further. Okay, fine. Let's move ahead. So we'll just first of all start the lecture, as I have just now told you that various cell fusion experiments, they actually led researchers to conclude that cytoplasmic factors can drive a cell cycle from one phase to another. Right? It means that a cell which is in a particular stage of a cell cycle, say for example in the G1 stage, and it has to enter the S phase. So it was seen that a cell that is in the early G1, say for example, if it is injected or fused with another cell that has actually entered the S phase of the cell cycle, it was seen that both the cells, they actually started the events of the S phase. That is, they both entered the S phase and started with replicating DNA. So it was then concluded that something was present in those cells which were in the S phase of the cell cycle, that they had something in their cytoplasm that led these cells, those which were in the G1 phase of the cell cycle, also to start the events of the S phase. This means in a normal cell, when a cell has started its cycle, it is in the G1, it has to just accumulate all those factors that are required for DNA replication, or you can say synthesizing those proteins in the cytosol, then only it can start the event of the S phase. So initial insights into this dependence of S phase on M phase came from cell fusion experiments of Poturau and Robert Johnson in the year 1970. Now, what they did was, I'll just explain this to you with the help of this figure, right? Instead of reading the text. Oh, so is this figure clearly visible to all of you? Okay, fine. So here you can see what these two researchers, they did was, they again, you can say they isolated cells which were in the different phases of the cell cycle, that is in the G1S, G2 and M phase. And what they did was they fused, cells can fuse and we can make a hybrid cell. So what they did was, initially they fused a cell which was in the S phase of the cell cycle, that is, its DNA was replicating, with a cell which was in the G1 phase of the cell cycle, and hence, you know, G1 phase does not involve replication of DNA. So it had a non-replicating DNA. They fused this cell, formed a hybrid cell, and when these cells were cultured in vitro, it was seen that both the DNAs were replicating. Now, you know, this DNA in case of eukaryotic cells, it has a nuclear membrane, as you can see, right? Since the nucleus were not fused, so it was very clear that nucleus was not containing any factor that could drive this G1 cell to start the events of the S phase. But since their cytosols were fused, it was only a cytoplasmic fusion, they just concluded that something in the cytoplasm of S phase cells led to the, you can say, the G1 phase cells also to enter into the S phase of the cell cycle and start replicating their DNA. Then, in the second set of experiment, what they did was they fused the same S phase cells with G2 phase cells, right? Again, in the G2 phase, there is no DNA replication, but G2 has one more regulation. See, now once a cell is completing its cell cycle for once, I mean, it is crossing all these stages, the cell also makes sure that the DNA replication should be only once per cell cycle, right? Because if the DNA is going to replicate multiple times, the cell will never be sure that how many copies of its chromosomes or DNA has been made and how to distribute them properly. So restricting DNA replication in the S phase to be only once per cell cycle is also a very important molecular aspect that the cells take care of while you can say regulating their cell cycle in a molecular way. So next what they did was they fused this S phase nucleus with G2 phase cells, right? And again, these S phase cells, when fused with G2 cells, it was seen that in the G2 phase, the DNA did not replicate. Now, it was very clear from the previous experiment that the S phase cells in their cytosol had that factor that could drive DNA replication. But then they concluded that in the G2 phase of the cell cycle, the cells actually accumulate those proteins that restrict the cell to replicate their DNA again. Because in the G1 phase, initial G1 phase, all this replication machinery is not present. But as soon as the cell completes the events of the G1 phase and enters the S phase of the cell cycle, it has already replicated its DNA. This means it has all the requirements complete that are required for it to replicate its DNA again. So this means as soon as they replicate their DNA once and enter into the G2 phase of the cell cycle, the G2 cells or cells in the G2 phase, they actually accumulate those cytoplasmic factors that restrict DNA replication again, right? This experiment is a very important experiment that actually led researchers to further know that what were actually those cytoplasmic factors that regulate this replication and non-replication of DNA. They drive the cell from one phase to another. See, this DNA replication, actually we take it as the center because this is considered to be the basic thing according to which the cell cycle is divided into different phases. So is this experiment clear to you? If you have any queries, please post your questions in chat, okay? And if you do not have any query, please type okay so that I can proceed forward. If you have any doubts, you can ask. Okay, Sharikul, can you please come over a microphone if it is possible for you? Okay, I'll explain it again for Sharikul. And in between, we have a question from Oindril, right? Oindril says, what if S cells were fused with G0 phase cells, right?
Hello.
This is a very quick question. Okay, Sharikul is over microphone. Yes, Sharikul, do you want me to
Ma'am,
explain the
can
complete thing again?
yes, ma'am. From starting, ma'am. The first one, ma'am. Explain once again.
Okay, please mute yourself. Okay, I'll take up Oindril's question also. This will help us to understand it again. I'll explain it to you again. So, okay, now this is a very interesting question from Oindril. What if S phase cells were filled with G naught phase cells? Right now, I want others also to post in chat your responses, and then I'll explain you again the whole thing along with answering Oindril's question. Andil, did I get your question right? Please type in chat. You just want to know what if G naught cells are fused with the S phase cells. Okay, first of all, Andil, this was an experiment that was conducted by two researchers, right? You have a question apart from this. We have not discussed the fusion of G naught cells here because that was not a part of their experiment, right? So first of all, are you clear with this thing? But this is a very good question that if we fuse S phase cells with G naught phase cells, now see, if a cell is in the S phase of the cell cycle, it must have completed all the events of G1 phase and then entered into the S phase of cell cycle. Now, whenever a cell is allowed to move from the quiescent stage of the cell cycle, that is the G naught phase of the cell cycle, into the G1 phase, right? What happens fundamentally is that in G naught phase, all those factors that allow a cell to complete the events of late G1 and enter the S phase of the cell cycle are restricted, right? So we have some external or internal factor that binds to those cells or stimulates those cells to complete the events of G1. So that all those proteins of the G1 and the S phase that were restricted for a cell to synthesize, they are now allowed to translate. Now, if we fuse these two cells, that cell will have all those factors, right, that can allow the cell to replicate its DNA and complete the events of those phases. So that is again going to drive those cells towards division and will allow those cells to proliferate. So Andril, are you clear with your question? Fine. So, okay, I'll explain again. Okay, please listen to it carefully because I can see a number of students have a query. Now you see you are reading this for the very first time and this is a very conceptual topic. So I'll just explain it to you again. Now, just I'll just go back and tell you, you just have to keep one thing in mind that cytoplasmic proteins, they decide the phenotype of any cell. If we talk about any living system also is general, whatever proteins it has in its cytosol decide what functions it is going to perform or what is going to be its phenotype. So cells, when they start their cell cycle, right, they accumulate all those proteins in their cytosol at different stages of the cell cycle. And a cell is not allowed to accumulate all these proteins all together because this process is very regulated. It has to be very coordinated, right? So in case there is accumulation of all these proteins very early, right, these cells phases will not be coordinated and it is going to create an issue. Okay, it can lead to an erroneous cell division. So the point is that if these cytosols were checked, the cytoplasm of these cells were checked at different stages of the cell cycle for the presence of proteins, it was seen that there was accumulation and deaccumulation of different proteins that led to the actual molecular events that need to be completed by the cell in that particular phase, right? So all these things were understood initially by this cell fusion experiment by these researchers, where what they did was they isolated cells at different stages of the cell cycle. That is, they stalled them in that particular stage. So initially, this was done to understand that the cytoplasmic behavior of all these cells in the different stages of the cell cycle is different. So what they did was, initially, they took an S phase cell and a G1 phase cell and fused them to form a hybrid cell. Now, the S phase, in the S phase, you know, it is called the synthesis phase, where a cell actually replicates its DNA. So it was obvious that in the nucleus of those cells, the replication has already initiated, and these cells are about to complete their DNA replication. But in the G1 phase of the cell cycle, the cell is actually, you can say, completing its growth and preparing itself for all the factors which are required for the cell to replicate. So the nucleus here is non-dividing. So they fuse the cells in such a way that the nucleus were not fused, right? So it was clear that it was not any nuclear behavior that was actually controlling this drive of a cell from one phase to another. And obviously, since their cytoplasm were fused, so it was very clear that during the S phase, there were some cytoplasmic proteins, right, that actually drive DNA replication. So once these cells were fused, those proteins were shared by both the nuclei, and they were easily transported through the nuclear pore complex to the nucleus, and they started DNA replication. So that is why both these nuclei, they displayed when grown further. I mean, this cell did not need to complete all those events because those factors were already present in the S phase, and both the nucleus were seen replicating. Sharikul, is this clear to you now? And Pooja, is this clear to you? You both had a question on this. Okay, so I guess it is clear to all of you. So again, now during the cell cycle, it is another important factor for a cell. Now, a cell has to just make one copy of its DNA during one cell cycle, because it has to keep one copy for itself, one set of its genome for itself, and give the other one to the daughter cell, right? So if suppose a cell divides twice, it is going to have three copies, right, of its entire genome, and then it will not be able to carry out the equal distribution of DNA to those cells. So it is very important, right, for a cell to also regulate that during one cell cycle, the DNA should be replicated only once. So in the second set, what they did was, because after S phase, what comes is G2 phase. So it was very clear that any control that could regulate the replication of a cell to once per cell cycle would be present in the G2 phase. So they then used the S phase cell with the G2 phase cell. And of course, they saw that in the G2 phase, there was no evidence of DNA replication in the nucleus. So when they fused those cells, DNA replication, in spite of having all the factors for DNA replication in the S phase, the G2 nucleus did not divide further. So this clearly indicated that in the G2 phase of the cell cycle, the cell actually accumulates those proteins, right, that restrict those proteins that drive DNA replication in the S phase and do not allow the division to, the replication to occur again. So is this clear to you all now, Sharikul and Pooja? The second experiment also. Okay, so I'll just proceed further. Then after these cell fusion experiments, various researchers, they just started working on what are actually these cytoplasmic factors which are present either in G1, S, G2 or M phase of the cycle that actually allow DNA replication or restrict DNA replication. So they just started working on that. What are actually these cytoplasmic proteins we are talking about, right? So these were called to be the regulators of cell cycle progression. So now we'll just see what these regulators are. And again, before that, we'll talk of the discoveries that actually help researchers to come to the conclusion and decide what these molecular factors are. So the cell cycle of all eukaryotes is controlled by a conserved set of protein kinases, which are responsible for triggering the major cell cycle transitions. Rhythmic fluctuations in the abundance and activity of cell cycle control molecules pace the events of the cell cycle. These regulatory molecules include protein kinases that activate or deactivate other proteins by phosphorylating them. These kinases are present in constant amount but require attachment of a second protein, a cyclin, to become activated. Right. So this is the gist of what was concluded. We'll again go back to the experiments which actually led to these conclusions. So after a lot of research, it was concluded that these molecular regulators actually involve some protein kinases. Kinases are proteins that can phosphorylate proteins and lead to activation or deactivation of many proteins, right? Like they can deactivate the replication machinery, or different, you can say they can lead to apoptosis if it is a faulty DNA replication, or they can initiate DNA repair. Plus, it was said that driving the cell from one phase to another was not regulated by just these protein kinases. These protein kinases are again, you can say, regulated by a regulatory protein which was called cyclin. So we'll just see now what were the experiments that actually helped researchers to identify the existence and functions of these two proteins to regulate the progression of cell cycles from one phase to another. And then we actually see what is the actual structure of this cyclin and these cyclin dependent kinases, and how they control and drive the cell into the different phases of the cell cycle. Levels of cyclin proteins fluctuates cyclically. Because of the requirement of binding for a cyclin, the kinases are called cyclin-dependent kinases, or CDKs. Cyclin levels rise sharply throughout interphase and then falling. Cyclin kinase, which is called MPF, corresponds to P in cyclin concentration. So now this complex together of cyclin and cyclin dependent kinase, it is called cyclin dependent kinase because it can only function and regulate the cell cycle if it is present in coordination with this, or found as a dimer with this cyclin protein actually. Now there are different families of cyclin dependent kinases as well as these cyclins in different type of cells or in different living organisms, right? And together, in general, it is called the maturation promoting factor. And why it is called so, we'll just see once we'll learn about the different experiments that actually led to the discovery of maturation promoting factor. So this MPF is said to be a dimer of CDC2, which is a type of cyclin dependent kinase, and the cyclin proteins, which is actually the regulatory protein that regulates the activity of this cyclin dependent kinase, or CDC2. So initially, 3 distinct experiments which were carried out by different set of researchers on different organisms, they led to the discovery of this MPF and the overall mechanism that how it regulates the eukaryotic cell cycle. So in the year 1971, 2 independent teams of researchers, which are mentioned here, found that oocytes arrested in G2 could be induced to enter the M phase. That is, the cells which have already completed the interphase can be allowed to enter the M phase by micro injection of cytoplasm from oocytes that have been hormonally stimulated, right? They just went to say that any cell that has already entered the division phase, that has crossed the interphase, if their cytoplasm was injected into a cell that is arrested into G2, right? Means it has completed the interphase, but it is not entering into division phase. It could be stimulated to start the events of the M phase. It thus appeared that a cytoplasmic factor present in hormone-treated oocytes was sufficient to trigger the transition from G2 to M in oocytes that had not been exposed to the hormone. Because the entry of oocytes into meiosis is frequently referred to as oocyte maturation, this cytoplasmic factor was named the maturation promoting factor, or MPF. MPF, then later on it was seen that it is not just restricted to oocytes or regulating the cell cycle of oocytes from the interphase to M phase. It was seen that it is also present in the somatic cells, where it induces entry into the M phase also. Okay, so we'll just move to the second experiment. Then the second major experiment was done in Saccharomyces cerevisiae, right, the yeast, where it was seen that there were some temperature sensitive mutants, right, that were not able to complete their cell cycle, and they had a defective cell cycle progression. And once the researchers, they worked at you can say molecular level, and they found that it was a CDC2 gene, a gene which was defective in those temperature sensitive mutants. I mean, due to high temperature, this gene was activated, and in these mutants, the cell cycle was enhanced. So then they concluded that this CDC gene is actually one of the you can say key cell cycle regulators, and its proteins, when synthesized and present in the cytosol, regulate the cell to complete the events of cell cycle. Then later on, a human gene related to CDC2 was identified and shown to function in yeasts, providing a dramatic demonstration of the conserved activity of this cell cycle regulator, right? So this CDC2 is actually that cyclin dependent kinase gene, right? And we'll just see the last experiment that was conducted. I'm sorry, there was a disconnection in between. So I was saying that just like yeast, a CDC2 gene was also identified in humans, and it was seen that if this gene is disrupted, its function is disrupted, the human cells were also not able to proliferate correctly and complete their cell cycle correctly. Then the last experiment that was important, it was done in the year 1983 by Tim Hunt and his colleagues. And then they actually identified that there are two proteins. Like I have told you already that MPF is a dimer of two proteins, these CDC2 and cyclin. So they actually identified in sea urchin and clam embryos that it is not one protein, because others were working on these proteins separately, and they could just work on either cyclin or cyclin dependent kinase. So Tim Hunt and his colleagues, they said that no, these were actually two proteins, and they actually talked of cyclins, right? The previous researchers, they talked of CDC, that was cyclin dependent kinase. Then they said no, cyclin dependent kinase or CDC cannot work alone. They then identified that there are other proteins called cyclins. Their accumulation and deaccumulation actually regulates the activity of these cyclin dependent kinases also, and coordination. So Hunt actually identified these proteins, the regulators of CDC or cyclin dependent kinases, these cyclins, and suggested that their periodic accumulation and destruction controls the entry and exit from the M phase. Then finally, in the year 1988, James Mallard, he actually purified this MPF, maturation promoting factor. Now see, everybody was clear. If there is a coordination between these two proteins, that is cyclin B and cyclin dependent kinase, the previous researchers were not able to purify or identify this protein in this dimeric form, that is the MPF, right? they could find either cyclin, they could find cyclin dependent kinase or CDC, right? I mean, they were not able to identify the correct stage, or they were not lucky enough to purify them, or you can say gather that information during the cell cycle when this protein, both these proteins were actually present as a dimer, and thus they form the maturation promoting factor. So this was done by James Maller in the year 1988, who purified MPF from frog eggs, and after its molecular characterization, okay, he revealed that MPF has some conserved regulator of the cell cycle, showed that this MPF is the conserved regulator of the cell cycle and is composed of CDC2 and cyclin B. So he proved that they come together also, and this cyclin actually regulates the activity of this cyclin dependent kinase. Cyclin B is regulatory subunit required for catalytic activity of the CDC2 protein kinase. Consistent with the notion that MPF activity is controlled by periodic accumulation and degradation of cyclin B during the cell cycle progression. Actually, researchers saw that this CDC2 or different cyclin dependent kinases, they were present in the cell throughout their life cycle. But it was actually this cyclin which was getting formed and destructed. See, cyclin dependent kinase can act as MPF, or it can drive a cell from interphase to M phase only if it has its regulatory subunit. It is just like a cap on the pen, right? So they said that if this cyclin is present and it forms a dimer with this CDC2, then only can this kinase be activated and further phosphorylate those proteins that can drive a cell into the division phase, right? So the best way the cell could regulate this protein was to destruct this protein when it does not want the cell to divide and allow the events of the interphase to complete. So this is what happens during the entire cell cycle. And you'll see not only just these two proteins form a dimer, there will be additional phosphorylation and dephosphorylation that after the formation of the dimer also are required, that can actually drive a cell from interphase to M phase. Okay. So did you get what is MPF and what are these experiments about? These experiments just tell that the cytoplasmic factor, which we are saying that regulates a cell from interphase to M phase, is actually the maturation promoting factor, and it is a dimeric protein of a cyclin dependent kinase. We have shown here CDC2 because these were the first genes that were identified after Saccharomyces cerevisiae in humans, and then cyclin B was the first cyclin that was initially purified and identified. Otherwise, in general, you can write down this is cyclin dependent kinase and this is cyclin. If you have any questions, please type in chat. Otherwise, I'll take you to the molecular control of the cell cycle. If you have understood these experiments, please type OK. Even if you go through this lecture again and you find something that is not understood by you, you can always ask this in the next class, because this molecular cell biology part, I mean the cell cycle molecular control, will run for 2 to 3 lectures. Okay, Andril is typing something. Andril, do you have a question? Okay, Sharikul, any questions? It is okay with Pranay, Pooja, Nutan. Okay, Sharikul has also typed in okay. I think he is not having any question. Now see, this topic is very, very important because I have told you, okay, we'll inject MPF into a non-dividing cell. Yes, these factors can initiate cell division because again, right, I have told you whenever a cell is not allowed to divide, right, this means there is some restriction that is not allowing the synthesis of all these factors. But give all those factors artificially to a non-dividing cell, we can stimulate those cells to divide. But you see, in nature, they are not allowed to divide, you can say further, right? So there is always an erroneous cell division because, of course, when they have been designed or destined to not divide any further, right? They have strong restriction signals, then those cells that are eventually allowed to divide when stimulated to do so. No, of course, I told you there are a lot of if and buts with this, right? There are a lot of things, there are a lot of other factors also that restrict this division. So we cannot just inject this into a cell that has the ability to not divide, right? That is a completely different part, right? That comes under health, and you could see that I mean, if these cells, they have very complicated systems, right? And if there is a damage in them, then only a cell can divide. You just cannot stimulate a cell to divide on its own, right? So they have very strong restriction signals as compared to other cells that, again, when allowed to drive through, lead to a number of errors. Okay, again is a separate molecular concept. Okay, Oindril, I think we should focus here and let others also understand the fundamental concepts. Okay, so now let's see how this MPF now actually regulates the cell cycle. I had shown you earlier yesterday a very simple figure of the cell cycle, right? I'll just draw it here for you. I'm not very good at drawing like this, right? You can just see we have had a very simple cell cycle. Okay, where this was the M phase, right? This is the G1 phase, this is the S phase, and this is the G2 phase, right? Now I have drawn this cycle again, which we have discussed today, or in this figure you can see this cycle is now drawn with respect to the maturation promoting factor, right? So now we'll start from here because this part describes the cell when it is in the G1 phase of the cell cycle, right? It is a newly formed cell, and you can see that cyclin dependent kinase is present throughout the cell cycle. But this cyclin B is missing sometimes, first of all, right? And then the next thing that you should see is that the you can say the structure of this cyclin dependent kinase with respect to these phosphates, these specific amino acids groups. Here in orange you can see phosphate groups attached. This behavior is also different for cyclin dependent kinase during the different phases of the cell cycle. So it is actually these two things that you have to keep in mind and know, that the behavior of cyclin B will vary in different phases of the cell cycle with respect to accumulation and destruction of cyclin B, whereas the behavior of the cyclin dependent kinase, which is shown here to be CDC2, will vary according to the phosphorylation and dephosphorylation of specific amino acid residues during the different phases of the cell cycle. So in the G1 phase of the cell cycle, this cyclin dependent kinase, right? It has three specific conserved amino acid residues, which are threonine 161, tyrosine 15, and threonine 14, right? These three amino acid you can say amino acid positions are said to be the regulatory positions, and phosphorylation and dephosphorylation at these amino acid residues can actually activate or deactivate this kinase along with binding to the cyclin B. So during the G1, the cyclin dependent kinase or CDC2 is present with all its you can say conserved amino acid residues which are in the non-phosphorylated state. So the cell grows, completes the events of the G1 phase, And as soon as it enters the S phase or is about to enter the S phase, it starts the synthesis of cyclin B, right? This cyclin B, once synthesized, right, there will be a number of cyclin dependent kinases and number of molecules of cyclin B. As soon as it is synthesized, it has an affinity for this cyclin dependent kinase. So it forms a complex with cyclin dependent kinase, right? This occurs, say, for example, in the S phase of the cell cycle. But now you see, this appears to be like MPF, but the cell has not started or entered into the M phase of the cell cycle. This means there must be some more addition, some more changes that actually drive this maturation promoting factor to be active. So these cells then completes the events of the S phase, that is, it replicates its DNA and then enters the G2 phase of the cell cycle. So during the S to G2 transition, right, there are special kinases that actually phosphorylate. The cyclin dependent kinase actually can autophosphorylate also all these three amino acid positions, right? It adds a phosphate to all these positions, right? So this phosphorylation now again does not render MPF to be active because the cell is still in G2. So then the researchers, they found that a dephosphorylation event actually occurs in the end of G2. Here you can see, right, that actually removes phosphates from threonine 14 and tyrosine 15. This dephosphorylation, right, is actually the switch that drives the cell from interphase to M phase. So as soon as this dephosphorylation occurs and this maturation promoting factor or this cyclin dependent kinase is bound to cyclin B with a phosphate attached at threonine 161, you can see this activates this cyclin dependent kinase, and this kinase actually phosphorylates all the important proteins that are required to by the cell to complete all the events of the mitosis, that is to complete all the events: spindle accumulation, breakdown of nuclear envelope, condensation of chromosomes. I'm not discussing that here because this is molecular control of cell division that we will take up separately. We have to take it phase wise, that what are the different proteins that are activated by this cyclin dependent kinase. So for here, you just understand that once this MPF is active, it actually leads to condensation of DNA, then breakdown of nuclear envelope by activating all those proteins that actually do these events for the cell. Now, the cell then enters the prophase, metaphase, anaphase, and as soon as the cell is in the end of the anaphase, right, it is about to complete its division, only telophase is left, the destruction of cyclin B starts, right? It starts because there will be multiple molecules of cyclin B, and the cell now actually prepares itself to exit division and again enter into the G1 phase of the cell cycle. So in the anaphase, destruction of cyclin starts, that completes by telophase, and by the end of telophase, all the cyclin B is destructed. If the cyclin B is destructed, then the cyclin dependent kinase cannot work alone, right? And all those proteins, they are not activated, and here this phosphate only at 361 is left, but still in the absence of cyclin, this kinase is not able to hold the cells in mitosis. So with the destruction of cyclin, complete destruction, cells exit. At that time, the cytoplasmic division is also complete along with the nuclear division. There are two new daughter cells which are actually the G1 phase of the cell cycle. So this figure basically explains how the cytoplasmic accumulation, deaccumulation, and alteration in the phosphorylation and dephosphorylation behavior of this particular cytoplasmic factor called MPF, a dimer of cyclin B and CDC2, actually regulates a cell to enter from one phase of cell cycle to another. I hope it is clear to all of you. It is easy to understand if you have clearly listened to the previous concepts, right? If you find it difficult, you can go through it once again and ask questions in the next class. But again, this is a very important topic, a very interesting and important concept that you should keep in mind. Okay, so I'll end the session now for today. And in the next class, we have talked about the cell cycle checkpoints, right? So we'll actually now talk about the molecular control of those cell cycle checkpoints and how the cells regulate to restrict their movement from one phase to another in case there is a damage in DNA. Okay, and then we'll also learn about what is the molecular control that regulates DNA replication to once per cell cycle. So we'll talk about this in the next class. Thank you so much. I'll end the session now.